Next up, Professor Mark Schnitzer, Professor in Biological and Applied Physics at Stanford University, who's going to present on optical imaging of large-scale neural codes and voltage dynamics in behaving animals. You're doing what I did to the last speaker, aren't you? Moving me off. Thank you.
Thank you. Many thanks to the organizers for the chance to speak with you today. I'm Mark Schnitzer, and my lab has been developing imaging techniques that allow us to visualize the large-scale ensemble neural codes and dynamics, and we're particularly interested in understanding how these underlie learning and long-term memory. Now, a main topic of discussion in my talk today will be the miniature digital fluorescence microscope that you can see there on the tip of a finger. This is 2 grams, small and light enough that a mouse can bear the microscope on its head. In this case, the mouse is actively exploring the arena that you see. Meanwhile, we can visualize the somatic calcium dynamics of about 700 hippocampal pyramidal cells in hippocampal area CA1. And I'm going to describe to you how we can use this technology to examine neural codes underlying memory. Now, when we consider what are the biological substrates of long-term memories in the brain, there are likely to be substrates at different levels of organization, at the synaptic level, the cellular level, and at the network level. In past research, neuroscientists have put a lot of work into uncovering what might be the candidate substrates at the synaptic and cellular levels, but those at the network level remain poorly explored, mainly due to lack of suitable tools. What we need are a suite of methods, a combination of methods that will, first of all, allow us to image cells that might lie deep in brain tissue, such as in areas pertinent to various forms of memory. We need to be able to track these cells over long periods of time, days and weeks, that are germane to the study of long-term memory. And we need methods that will allow us to see the activities of large ensembles of cells in freely behaving mice or other animals during natural forms of behavior. And what I'm going to show you today is that by combining these methodologies, we have new opportunities for performing long-term tracking studies in which we follow the evolution of neural codes and dynamics during learning and memory retention. So the first element, the ability to see cells, we developed a number of years ago, micro-optical needles, or microendoscopes, as seen here. We now have about 200 different designs of these. The one on the left you see is 1,000 microns in diameter. The one on the right, 350 microns in diameter. At the tip of each, you can see a small micro-optical objective that gives us the micron scale resolution we need to see cells in deep-lying tissues. These probes can be chronically implanted directly into brain tissue. So here we have some examples in mice that are expressing a fluorescent marker in a subset of the hippocampal pyramidal cells. And we can follow these individual neurons, in this case over 2 months. And if you look at the middle column, you'll see a close-up. We can follow individual dendrites in these fluorescent neurons and come back to them repeatedly with high mechanical precision. By choosing the appropriate microendoscope with diffraction limited resolution, we can even resolve the individual synapses of these cells, seen here by way of their postsynaptic morphological entities on the synapses, the dendritic spines. And we can track these individual synaptic elements over weeks, as seen here in this 3-week experiment. Some of the spines persist throughout, others arise midway through the experiment, and still others disappear midway. And so this gave us an opportunity to examine collectively the microscopic kinetics of these synaptic elements. And in a recent work, we found that their turnover kinetics here in the hippocampus is consistent with the previously reported timescale for the duration of hippocampal dependent memory in tasks, for example, that involve spatial or episodic memory. So we have a concordance between the microscopic timescales and what manifests at the behavioral level. Now, if we want to understand how large networks of such cells work together, ideally we want to be able to track their collective dynamics in freely behaving animals, and I'll be focusing here on freely behaving mice. You already saw the miniature microscope that we used to do that. It has multiple virtues. It allows the animal to behave freely and is compatible, therefore, with the vast majority of behavioral assays that are commonly used in rodent neuroscience. And we have a broad field of view. We can sample from up to 1,000 cells or more, as I'll show you momentarily. This is the small microscope, the digital fluorescence microscope, 2 grams, based on semiconductor optoelectronics. And my colleagues and I have started a company, Inscopics Inc., which has now commercialized this approach for the benefit of neuroscientists and neuroscience research, and it's presently being used on 3 continents. If you take a look here at what the components of the microscope are, each mouse is carrying 2 printed circuit boards. There's a cell phone CMOS camera board at the top, an LED illumination board at the side. There's also a dichroic and filter set, miniaturized objective peeking out. This device moves between the different mice that you might have for study, whereas the microendoscope would be chronically or permanently implanted in the tissue of interest. And this was the fodder for 3 different PhD dissertations and was a close collaboration with my colleague Abbas Al-Gamal in Stanford's electrical engineering department. Now here's what it looks like when a mouse is wearing the microscope. The animal can behave naturally, it can move at high speeds. Meanwhile, we're streaming digital brain imaging data right into the USB port. So this is our computer mouse, if you will. Now this is what a good afternoon looks like. We can have multiple mice plugged in with this technology. We can be tracking the large ensemble dynamics, hundreds of cells per animal. And in a single afternoon, we can gather several terabytes worth of brain imaging data. So we have very good statistical power now for computational studies of large-scale neural coding. Now, combining all the different approaches that I've been describing, Yaniv Ziv, who now has his own group at the Weizmann Institute in Israel, and Laurie Burns, one of the inventors of the miniature microscope, conducted a first study in which they tracked the hippocampal representation of space over many days, in fact, multiple weeks. So you've already seen the kind of data that the combination of the endoscopes and the miniature microscope can produce, and this is further attained by targeting a genetically encoded calcium indicator to the CA1 pyramidal cells in this portion of the hippocampus known to be required for spatial navigation and spatial memory. From that video, we can extract computationally about 700 hippocampal pyramidal cells, seen here by way of their red cell bodies on top of the green fluorescence background image. The latest version of the microscope has a broader field of view. We can visualize about 1,000 neurons at a time. This is our best mouse ever. So we got, in this mouse, 1,202 CA1 pyramidal neurons in a single imaging session. And I would say this is not typical. 1,000 in hippocampus would be more typical. Now, it's been known for a long time in the hippocampus that the pyramidal cells have place coding properties. They preferentially fire when the animal is in one particular portion of its arena. We can recapitulate this now optically. So to illustrate, there are 2 different arenas here, seen from a bird's eye view, a square box and a circular box. The heat maps indicate the cell's probability of firing when the animal is in a particular portion of its arena. So if we take cell 348, for example, you can see it preferentially fires in the south of the square box, but in the northeast of the circular box. So these kind of properties had been seen before electrophysiologically, but now we can visualize them optically in real time. So here, for example, is a mouse that Yaniv and Laurie trained to move back and forth in this linear track. Take a look at that cell that I've circled there. You'll see that it characteristically fires when the animal is in a particular portion of the track, and I've noted that with the arrow. You'll also see that the mouse has to be going from top to bottom, not bottom to top. So whenever you see that cell fire, you know exactly where the mouse is in the track, and you know in which direction it's headed. And therefore, by extension, you might imagine that if we could follow the activities of all the neurons under view, we might be able to reconstruct the animal's entire trajectory. And I'll show you in just a moment that we can do exactly that. Now, when we analyzed these data, after having imaged the mice every 5 days for over a month in that track, we found that there was a complete map of space across the month. Now, in these plots, the track has been rotated 90 degrees, so it's lying horizontal, and we've configured the cells with their representative place fields at one end of the track to be at the top of the plots, and the cells that preferentially fire at the opposing end of the track are organized at the bottom of the plots. And what you'll see, there's a complete map of space there across each day of the experiment. However, if we focus our attention on the set of cells with statistically significant place fields on the first day of the experiment, and we follow their map over time, we see that the map gradually fades out. On the other hand, if we look at the set of cells with statistically significant place fields on the last day, and we look retrospective, you can see that map gradually forming. Now, if you look carefully, you'll see that these maps are fading and forming in a very particular way. They're not smearing out, they're retaining the crispness of their spine, but rather they're losing cellular membership. And that observation is further quantified here. What we did was we tabulated lists of cells with statistically significant place fields on each day of the experiment, and we compared the overlap in those lists. And between any 2 pair of days that you might choose, it was about 25%. It actually was more like, it was 25% at closer days, I should say, and gently declining to something like 15% if you took a pair of days that was separated by the full 30-day interval. However, despite this turnover in the cells that were involved in a particular day, if a cell came back to the place code, it nearly always encoded for the same location on the track. And you can see that on the right. These are pairwise histograms across all pairs of days of cells' displacements in the centroids of their place fields when they return to the code. And these are all sharply peaked at the origin, indicating that when a cell does come back to the code, it nearly always represents the same location on the track. So we like to describe these data by saying that the place fields of these cells are spatially invariant, but temporally stochastic from day to day. So I like to make an orchestra analogy. Imagine that you went to the orchestra every 5 days for a month. Each time you go, they're playing Beethoven's Fifth Symphony. But who's playing the cello might rotate from day to day. Sometimes it's Bob, sometimes it's Alice. But if you see Alice on a given day, she's always playing the same instrument. Now, as I mentioned earlier, we can take the brain imaging videos and actually reconstruct the animal's passage through space. Lacey Kitch, a computational engineer in the lab, developed a Bayesian decoding algorithm that could do exactly that. The black traces are the mouse's actual passage back and forth. The red is what you would estimate from the brain imaging video. Now, despite the turnover that's occurring at the level of the individual cells, at the ensemble level, these decoders persist in their quality for about a month. So if you look at the last row, Lacey has taken the data from a month ago, used that to build the decoder, and then applied it to today's imaging data set, if you will. And you can see there is some decline in the fidelity of the reconstruction. Nevertheless, it's highly statistically significant, indicating that at the ensemble level, the map of space is persisting for about this month-long time period. Now, in more recent work, Yaniv and Lacey have been asking the animal to do a spatial memory task. This is an 8-arm radial maze. One of the arms contains a nutritional reward. The animal is randomly placed in one of the other 7 arms, and it has to use distal cues on the wallpaper that are not visible in this video to remember the spatial location of the reward and to navigate to it. Meanwhile, we can visualize the ensemble dynamics in the CA1 hippocampal network. Okay, now in the closing moments, I want to give you some glimpses of new techniques that may be coming over the horizon to help us study problems of the kind that I've been illustrating. Everything I showed you so far made use of calcium imaging as the way of looking at neuronal dynamics. What can we do in the years ahead? Well, one thing we definitely would like to do is to be able to visualize how different brain networks in different areas may work together. This was something that David Anderson mentioned in his talk. Well, we can implant more than one endoscope into the brain tissue. And we can use the fact that microendoscopes can be packed densely to interrogate more than one brain area at a time. So as an initial attempt at this, Jerome Lecoq, who's now at the Allen Institute, constructed a double-armed microscope. The arms were flexible, allowing him to interrogate 2 brain areas of his choice. So here are some examples: motor cortex and somatosensory cortex on the left, 2 different visual cortical areas on the right, so the areas can be nearby or further apart. Since they're microendoscopes, we can either use them toward the brain surface or in deeper tissue. So here's a pair of images acquired in hippocampus and in the frontal cortex. I'm going to show you what this looks like in a head-fixed but behaving animal. You'll see the animal is viewing a video monitor, and as the visual stimulus begins, we see cortical activity in these 2 different visual areas simultaneously. And then using the kind of computational techniques I illustrated earlier for the hippocampus, we can extract the individual cell bodies, in some cases some of the dendrites of these cells, and begin to analyze how the networks in these 2 areas may work together. We'd also like to get better time resolution. So calcium imaging has some limitations. What about imaging the transmembrane voltage dynamics of neurons directly? And so in recent years, there's been rapid progress in the development of genetically encoded fluorescent voltage indicators. In our lab, this work has been spearheaded by Yiyang Gong, who now has his own group at Duke University. And he's been developing Fret-opsin indicators that are work by a protein fusion of a voltage-sensitive protein to fuse to a very bright fluorescent protein. Here are some examples of indicators of 2 different colors. We've recorded from these neurons electrically and optically, and you can see we get a very faithful report with the optical signal. This works by using a protein from the archaeorhodopsin family of proteins. It has voltage-sensitive absorption. And so when the neuronal membrane depolarizes, this molecule becomes a better absorber of light. It's a very dim fluorophore, and this is why we have to couple it to a very bright genetically encoded fluorescent protein. Now, we've shown recently in awake mice that this indicator gives us single spike sensitivity. So dual recordings using loose patch electrical recordings and optical recordings have shown us that we can catch nearly every action potential in an awake mouse. And moreover, we can do so with about 200 microsecond spike timing precision. So if you see the panel in the lower right, this shows us a very nice Gaussian distribution of the spike timing errors with a width of about 200 microseconds. Moreover, we can not only see spikes, we can also see subthreshold facets of the membrane voltage dynamics. This is illustrated here in recordings actually from a neuron in the intact fly brain. You can very nicely see the action potentials as well as subthreshold facets of the depolarization. So are we there yet at being able to accomplish the kind of imaging studies I showed you in freely behaving animals, but with voltage? Not quite. We're doing them in awake animals, but head fixed, and we think that we're going to be able to take the additional strides necessary in the years ahead. So with that, I'll sum up. I mentioned numerous lab members as we went along. These are additional people that contributed to the experiments that I described, and I'd also like to close by thanking the funding agencies that made this possible. Thank you.